Current autologous therapy is personalized medicine at its most extreme. You harvest T cells, you ship them, you edit them, you grow them for weeks,
Yeah.
then ship them back.
And that waiting period is clinically dangerous for these patients. And if the harvest fails, you're back to square one.
So the whole industry is looking at UCAR-T.
It has to. This uses allogeneic cells, so cells from a healthy, non-patient donor. You manufacture them in huge batches, store them, and they're theoretically ready to go, ready to infuse the moment a patient needs them.
Now, this is where the real tech challenge comes in, right? When you introduce foreign cells, you have two major rejection problems you have to solve with gene editing. The first is GVHD,
Graft versus host disease.
the donor cells attacking the patient.
Right. And the second problem, which is often overlooked, is host versus graft rejection, or HVGR. This is where the patient's own immune system sees the donor cells as foreign and just wipes them out before they can work.
Okay, so if the T cells are the guided missile, GVHD is the missile attacking its own base, and HVGR is the base shooting down its own incoming missile. We need to disable both.
That's a great analogy. And to disable GVHD, we rely on precision gene editing, usually CRISPR, to essentially blind the donor T cells. We knock out the gene for the T cell receptor alpha constant, the TREX gene.
No track, no vision. The cell can't see the patient's tissues to attack them.
Exactly. But what about HVGR? Let's look at how innovators are tackling the patient's rejection of the therapy. We saw a powerful example from Northang Bio, their CTA-101 product.
What makes CTA-101 so significant?
Well, it was the first domestic UCAR2 innovative drug approved in China based on CRISPR editing, and they have this fascinating double knockout strategy to make sure the cells persist.
So they handled GVHD with the trait knockout. What was the second knockout for HVGR?
They also knocked out the CD52 gene. This is really key. Knocking out CD52 lets clinicians give an anti-CD52 antibody as part of the pre-treatment.
And that antibody would normally kill the CAR T cells too.
Right, but because the CD52 target has been edited out, the antibody suppresses the patient's host immune system, preventing that HVGR response, but it leaves the CTI-101 cells totally untouched.
So they made the therapy invisible to the drug that's suppressing the host defenses. That is a highly sophisticated strategy to give the CAR T cells time to work.
And the early results seem to validate it. In a trial of 6 heavily pre-treated adult B-ALL patients, a really tough population, they reported an 83.3% complete response rate.
Wow.
That's a huge data point. It proves an off-the-shelf product can navigate that GVHD-HVGR tightrope in the real world.